Well, thank you, Dr. Clark, and thanks to all of you for coming this morning. It's really a pleasure to be part of this Grand Rounds series here and to tell you a little bit about some of the work my group and our colleagues have been doing using this sort of unexpected ally here, the zebrafish, as a new approach for discovering therapeutics. Want to just start with some disclosures and appreciation for funding. Most relevant for the things I'm going to be talking about today are Kairos Therapeutics, which has licensed some of the compounds I'll talk about, and then support from the NIH, American Heart Association, and Merck for some of the projects I'll talk about today. So I thought I would start off with a fairly silly rhetorical question, which is outlined on this slide here. It's sort of a thought question. Where would you rather be a patient? In a 2017 hospital with a 1950 pharmacy, or in a 1950 hospital with a 2017 pharmacy? Now, I came from Mass General Hospital, and so I used to present this question to different clinical groups. And once I was at a competing hospital, and someone in the back row, when I talked about a 1950 hospital with a 2017 pharmacy, someone in the back row yelled, "That's where you work!" But obviously, there's no right or wrong answer to this question. But if you think it's even close, that suggests that the advances we've had in the last 60 or so years in pharmaceuticals have been equally important to all the advances in diagnostics and procedural medicine. And I think as we look forward to the next 50 years, advances in pharmaceuticals will likely continue to be a major driver of improvements in human health. And so I've been spending a lot of time with our college thinking about the question of what role the College of Pharmacy should be playing in that process over the next 50 years, and what role should the University of Utah and the health system in general be playing in all of that. Obviously, historically, the roles between industry and academia have been fairly well defined. Academia has been focused on publishing fundamental discoveries and carrying out clinical trials, whereas industry has largely taken the lead on almost everything else, from choosing which diseases they would tackle, to picking the therapeutic targets, doing the actual process of small molecule discovery and optimization, the preclinical development and clinical trial design. And so that obviously leaves much of the important decision-making in the hands of industry. And I think that there's a really important role, especially moving forward, with academia taking a greater and broader set of roles, including not trying to replicate what industry is doing. They'll probably continue to target large markets and use their conventional approaches to drug discovery. But I think academia can be doing important things to target tractable problems, even if they aren't huge market diseases, and to develop unconventional and creative approaches discovering drugs. And this, I think, is particularly important as we realize that there are actually thousands of rare orphan diseases that are not being touched by the pharmaceutical industry. Although these are small diseases in singletons, in aggregate, they represent a huge percentage of all of disease. And so we need to find ways of developing therapies for these orphan diseases. So just a little bit about my personal history, how I became excited about the prospects of doing small molecule discovery in academia. And I think it will just illustrate a little bit about the approach that we're taking. My conversion to this approach began more than 20 years ago when I started grad school at Harvard. And I was assigned to study this chemical compound I'm showing you right here. I guess my pointer isn't, there we go. Rapamycin is the name of this compound. It had been extracted from a soil sample on Easter Island, and not much was known about it, but it had been shown to have a transforming effect on cultured cells. So if you dumped it on cultured cells, they would change their phenotype. Specifically, they would arrest in a specific stage of the cell cycle. And so our lab had wanted to try and understand how that worked. And so a grad student who was a couple of years ahead of me, had taken rapamycin, attached it to some beads, ground up a bunch of cow brains, and basically poured them over this column and pulled out a protein that bound to rapamycin and sequenced that. And this was a protein that is now called mTOR, or the mammalian target of rapamycin. Since then, I and others were able to study the functions of mTOR with this small molecule, and it became a really powerful tool for rapidly disrupting the function of mTOR and seeing what impact that had on biological systems. And since then, there have been tens of thousands of studies using rapamycin to study mTOR's function, and we now know a great deal about how mTOR functions. Interestingly, rapamycin has also gone on to become a clinically used compound for, as you probably know better than I, as an immunosuppressant after organ transplant and as a coating on drug-eluting stents in the heart. And so this really got me excited about the idea that you could discover small molecules that cause a change in phenotype in a cell or in an organism, and then you could use that to learn new biology and, in some cases, develop new therapies. Now, this approach of using phenotype to discover a small molecule is really kind of an old-fashioned or classical form of drug discovery, where you start by picking the phenotype. You then screen for compounds out of a collection that can perturb that phenotype, and maybe only toward the end of the process do you understand what the target is. This old-fashioned way of discovering drugs has given way to this more modern approach, a target-based approach, which is really dominant in industry now, where you pick a target based on what you've read in the literature and what you speculate might modify disease phenotype. You then create an in vitro assay, you screen thousands or millions of compounds, you find those that bind to the target, and then only at the end of the process do you test the effect on the phenotype in animals or humans. So this works well in some cases and has become the dominant way of discovering drugs. In fact, Swinney and Anthony a few years ago looked at all of the drugs that have been discovered since the 1980s, and they found that about 95% of all expenditures in the pharmaceutical industry have been on this 95% of target-based efforts, and there's only a residual 5% of effort using phenotypes to discover drugs. What was really surprising and interesting is that when they looked at where the new drugs were coming from, this is what they found. They found that actually about 2/3 of all the new drugs that are coming out in these first-in-class drugs were coming from that little sliver of phenotype-based approaches. And I think this really startled a lot of people, and especially in the pharmaceutical industry, and helped them realize that maybe we shouldn't be abandoning phenotype as a way for discovering new compounds. So why has phenotype-driven discovery outperformed target-driven discovery? Well, I think for many diseases, we just don't know what the optimal targets are. There are probably many diseases that you study that we don't understand enough to know what the optimal therapeutic target is. In other cases, optimal therapy requires perturbation of multiple targets. So if you really need to perturb multiple members of a family, for example, to have a therapeutic effect of a protein target family, then you can't easily do a high throughput screen around multiple targets. And then I think the final reason is that in vitro discovery fails to predict in vivo effects, both good and bad. So often when you start out with a target and you find a compound that modifies that target in vitro, it's hard to predict the good and bad effects that will happen when you move that into an animal or into a human. And so what my lab has been building over the last maybe 15 years or so has been an approach where we're trying to use zebrafish as a screening platform where we can screen large chemical libraries and find those that create phenotypes that are of interest to us. And the hope is that then we'll be able, like in the case of rapamycin, to use those small molecules to discover their targets, learn some new biology, and in some cases even develop new therapies. So why zebrafish? Well, I think you know that there are many zebrafish researchers here at the University of Utah, so hopefully you know something about this model organism. But one of the really compelling things about it is that it's so genetically similar to us. 82% of disease-causing genes in humans have an analogous gene in zebrafish. And also, it's important that they're vertebrates, and so they have most of the same organ systems that you and I have and very similar physiology. So unlike fruit flies or worms or yeast, these other genetic model organisms that are common in biological study, fish actually have livers and pancreases and hearts, et cetera, et cetera. Another important advantage, especially for drug discovery, is the advantage of scale. So we can house tens of thousands of adult zebrafish in relatively inexpensive facilities where we just really only need water and food to keep them going. And they're very prolific. Each female can lay about 200 eggs at a time, which enables you to produce literally tens or hundreds of thousands of zebrafish embryos a day. And they're also quite small. What you're looking at here is a single well of a standard 384 well drug screening plate. So this is a dish that's about the size of your cell phone with 384 chambers in it. And you can see that these fish can just sit in water and live in there for days, just surviving off the yolk that they have. And so zebrafish researchers have developed all sorts of phenotype-based assays where in very high throughput, they can assess things like vision and hearing, cardiac contractility, insulin and glucose regulation, learning, tumor metastasis, gut motility, healing, regeneration, and many other kinds of medically relevant phenotypes. And we, over the last decade or more, have been able to apply this approach of high-throughput screening in zebrafish to a variety of different disease-relevant phenotypes, ranging from vascular occlusion down to schizophrenia. And here I'm showing you, again, a 96 well plate. In this case, we have about 8 or 10 zebrafish in each well of this 96 well plate that's the size of your cell phone. Some of these compounds discovered by us and by others are in clinical development. The most advanced of these compounds are in early phases of clinical trials, and then there are many others that have been validated in mammalian models and are in preclinical development right now. So what I hoped to do today is share with you 3 fairly superficial vignettes of how we've applied this paradigm in 3 very different areas. One around a bone disorder called heterotopic ossification, and describe a compound we discovered called dorsomorphin. I'll then tell you a little bit about discovering compounds that protect the heart from chemotherapy and a compound called visnagin that we discovered. And then finally, a little vignette that's topical, a newer project in our lab about modeling opioid self-administration in zebrafish. And I realize these areas may not be of specific clinical interest to you, but hopefully they give you a sense of how you can apply this kind of approach, and maybe it will spark some ideas in your minds about how the specific diseases of interest to you could be adapted for drug discovery. And I'd be very eager to talk to any of you that have ideas about how that might happen. So the first little vignette is about heterotopic ossification. This is where ectopic bone forms places where it shouldn't. It's common after trauma, burns, and surgery. And often there's a involvement of a class of proteins called the bone morphogenetic proteins in many of these forms of HO. One of the most interesting and profound forms of heterotopic ossification is fibrodysplasia ossificans progressiva, FOP, showing you some photos here. This is a rare disease where muscle and other soft tissues ossify. So these kids are born almost imperceptibly affected. They have a slight defect in their great toes, which are pointed a little bit inward. Otherwise, they look completely normal, and often the diagnosis is missed at birth. But within a few years, they start to develop bony lumps under their skin. Sometimes their attempts are made to surgically resect these growths, which causes increased inflammation and often an exacerbation and acceleration of the heterotopic ossification. Usually within a few years, these kids become locked in as they lose mobility of their joints, their jaw. Even the intercostal muscles begin to ossify in their diaphragm, and they often die of restrictive airway disease. So it's a really horrible and rare disease. Only about 1 in a million children have this disorder. There are no effective treatments. And it was discovered about a decade ago that it's caused by activating mutations in a protein called ALK2, which is a receptor for these bone morphogenetic proteins. And so we basically knew that this is a normal zebrafish embryo here, and we knew that genetic manipulation or activation of the BMP pathway caused ventralization of zebrafish, where you get an expansion of these ventral tissues and loss of the dorsal tissues. And we were able to perform a screen of thousands of chemical compounds in 96 well plates, looking for those that could dorsalize the embryo. And what we discovered was a compound out of several thousand that we screened that we called dorsomorphin because of its ability to dorsalize the embryo. So here again is a normal zebrafish embryo. The head is the dorsalmost tissue, and this ventral fin of the tail is the ventralmost tissue. And what we found is that by adding the compound at different times and different doses, we could get progressively more strongly dorsalized animals. So here, for example, we have just loss of the ventralmost tissue, and then here more and more profoundly dorsalized animals. This looked, for all intents and purposes, just like what happens when you inhibit or genetically mutate ALK2. And so we hypothesized that our compound might be inhibiting ALK2. And through a series of biochemical studies that I won't show you, we were able to confirm that, in fact, this is dorsomorphin here, bound in an X-ray crystal structure to ALK2, that same gene that's mutated in kids with FOP. And so we then did some chemistry in collaboration with Greg Cooney and his group, and we were able to take dorsomorphin and improve its potency considerably to the point where we had a really good tool compound for testing the validity of this approach in mice. And so our collaborators, Paul Yu and Ken Block at Mass General, we were able to develop a mouse model in which we were able to activate that human form of ALK2 present in FOP just in one of the hind limbs of a mouse. And what we found is that 15 days after birth, the gastrocnemius muscle here was starting to ossify. And by 30 days, you can appreciate there's massive ossification of all the soft tissues here in this hind limb. When we treated with this dorsomorphin derivative, we found that we could dramatically decrease the heterotopic ossification. And you can probably see that here in these videos. These are the FOP mice. Again, one hind limb has is expressing that mutant form of FOP ALK2 gene, and you've completely lost use of that hind limb. And then this is its sibling that's been treated with LDN compound, and you can see some lag in that leg, but it's retained use of that limb. At that point, we were able to partner up with an NIH program called Therapeutics for Rare and Neglected Diseases. This is a group of people mostly with pharmaceutical industry expertise, and they've been able to contribute to the following steps in preclinical development: a lead compound selection through medicinal chemistry. This is now over 1,000 derivatives of dorsomorphin that they've synthesized and characterized, and done all sorts of toxicity studies, PK studies, to the point where we've now developed derivatives of dorsomorphin that are highly potent and selective for ALK2 relative to the other members of the ALK family, and that have good pharmacokinetic properties. Here was that original LDN compound that we tested in the mice. You can see it has a relatively short half-life of about 3 hours, and we have derivatives now with much longer half-lives. And so we've now developed a whole family of these compounds that have the properties that you might want for a clinical candidate. And so we've now licensed these compounds out to 2 companies, Keros Therapeutics and La Jolla Pharmaceuticals, and they're developing these things hopefully for a clinical trial within the next year. There's some real challenges in clinical development of these kinds of compounds. The first is that there are so few FOP patients, only about 2,000 in the world. It's also a progressive disease with pediatric involvement, so that makes it really challenging to do long-term exposure studies. But there's such a compelling connection between the drug mechanism of action and the disease mechanism that we really think this is a trial that needs to be done. And we're thinking that shorter trials can be designed around planned interventions. So you can imagine surgically going in and trying to release the jaw or a limb, and then adding in the compound for a brief period of time to see if you can prevent reossification. So the second little vignette I wanted to share is in a completely different area. In this case, it's a cardiovascular area. And this story really starts with this molecule I'm showing you here, which is called doxorubicin. Many of you are, I'm sure, familiar with it. It's one of the most potent chemotherapy drugs. It remains a mainstay of clinical therapy for a wide range of different tumor types. One of the main limitations of this drug, although it can be very effective, is that it's highly cardiotoxic. In fact, you have to monitor lifetime dose of exposure, and doses over about 400 milligrams per square meter cause very high rates of heart failure in people who receive doxorubicin. It's not entirely known why the compound is so cardiotoxic. It's believed that the mechanisms of damaging tumor and damaging the heart are different. In the case of rapidly growing tumors, it's thought that inhibiting DNA replication is a major contributor. Whereas in the case of heart, a number of mechanisms have been speculated, including production of reactive oxygen species, but there's some hope that you might be able to find compounds that block the effects of doxorubicin on the heart without impeding its ability to be effective as an anti-cancer agent. A postdoc in the lab named Yan Liu developed a zebrafish model of doxorubicin-induced heart failure. And basically what he did was treat these zebrafish at an early stage. You can see, sorry that my pointer isn't working very well here. But you can see on the left here, the arrow is pointing to the heart of the zebrafish. These embryos are so transparent that you can just look in a microscope and see the heart beating. When you treat for a few days with doxorubicin, you see something like shown on the picture on the right, where you have a large pericardial edema and the heart is beating weakly. Down at the bottom, you can see something that may look like an M-mode echocardiograph. This is done just with light microscopy, where we take a video of the heart, draw a line of interest across the ventricle, and then you can see over time the contractility of the heart, where you can measure the diastolic and systolic diameters and measure a fractional shortening. And after doxorubicin treatment, you can see a reduction in the fractional shortening from about 50% down to about 15%. So pretty severe heart failure phenotype in these animals. Yan then did a screen where he looked at several thousand compounds, looking for those that could revert the doxorubicin-treated animals back to a normal phenotype. Oh, thanks so much. Yeah. And after screening several thousand compounds, Yan found these two, this one called visnagin and this one diphenylurea, both of which could produce 100% rescue of both the contractility and blood flow phenotypes in these animals. And so, for example, this is the doxorubicin-treated fish. These two have been treated with doxorubicin, but also with visnagin or diphenylurea. Hopefully you can appreciate that the heart looks much better here than here. And when we quantify that by this sort of M-mode imaging, you can see that we can get a complete rescue of the function of the heart. So, of course, that's only in fish, and then the first thing you want to do is figure out whether that effect is conserved in mammals. And so we went to the mouse, and we looked at several different models. The one I'm showing you here is one that's thought to be most similar to what happens in the humans. And this is a model where for 5 successive weeks, you give them a dose of doxorubicin, and then you wait 12 weeks and use echocardiography to quantify the function of the heart. You can see dox treatment causes a reduction in fractional shortening and strain rate, which can be rescued by co-treatment with visnagin. Obviously, if you protect the heart but you protect the tumor, then you're not helping anybody. And so we needed to test whether these compounds were protecting the tumor. In collaboration with Dan Haber's group at MGH, we used a human breast cancer cell line that's been xenografted into the mammary fat pads of mice. The tumors are allowed to grow until they reach a consistent size, and then they're treated, either untreated, in which case they continue to grow, treated with doxorubicin alone, in which case you can reduce the tumor size, or with doxorubicin plus visnagin. And we found that visnagin, although it can protect the heart, does not protect the tumor. So, as in the story I showed you with FOP, we then turned to chemistry to try and make even better versions of this molecule. And we now, the details here are not so important, but you can see that we now have synthesized and tested analogs that have very high potencies. Here at 1 nanomolar, these compounds are able to protect zebrafish hearts. And when we test those derivatives in mice, we find they're also incredibly protective. The lowest dose we've managed to test so far is 0.1 milligram per kilogram, which is quite a low dose. And even that low dose has a very profound protective effect in mouse hearts, as shown here by quantifying the amount of annexin staining, or the number of apoptotic cells in the heart after doxorubicin treatment. So, shifting gears one last time, moving to an area that's been of interest to us just recently. Some of us in the lab have been really troubled by the alarming increase in opioid overdose deaths in the U.S. As you can see from these plots from the CDC, there's no evidence that we've got this under control, and in fact, this seems to be just exploding on us right now. As you likely know, there are about 40,000 people overdosing and dying in the U.S. This year, but that's only the tip of the iceberg, and there are actually several million who are struggling with opioid addiction. There are very few medical treatments available. The ones that we have are those that affect the opioid receptor pathways, either by replacing heroin with slower acting opioids like methadone, by blocking the opioid receptor with compounds like naloxone, or by using partial agonists like buprenorphine. Some of these have some beneficial effect, but clearly insufficient to stem the crisis that we're faced with right now. And so we started asking ourselves whether we could use the zebrafish to find a way to contribute to the search for medical treatments for opioid addiction. There had been some foundational work that suggests that zebrafish respond to opioids. These were passive or non-contingent assays where zebrafish were in a tank and opioid was added to their water, and you could measure physiological effects on the fish, and that you could also show that with time they developed a preference to be in a location where they had previously received an opioid. So those kinds of non-contingent experiments are really suggestive, but what you really want to be able to measure motivation for drug seeking is a contingent assay where there has to be some active operant behavior performed by an animal to seek the drug. And so Gab Bossé in the lab, shown here, decided to build an arena where zebrafish could self-administer opioids. And he did this all with off-the-shelf parts for just a few $100s. Basically, he built a plastic box where water would flow in one side and out the other. And in that arena, he then could place different platforms, in this case showing a white platform that he calls the inactive platform and a yellow platform, the active platform. And he put small cell phone cameras above each platform to record the number of times that the fish would swim over that platform. And if a fish swims over this inactive platform, that event is recorded, but nothing happens. But if the fish swims over this active platform, that event is recorded, but also a green light flashes and a small pump delivers a dose of opioid to the platform. All of this was tied together with all the motion tracking, dosing, and analysis was done with a small, inexpensive computer called a Raspberry Pi, and with software that was custom written. And what Gab did then was to take groups of fish, usually about 15 fish, and just place them in these arenas for 50 minutes a day over 5 days. And when the naive fish go into the platform, they swim around and they may accidentally swim over these platforms as they're exploring. And Gab recorded the number of times that they swam over these individual platforms. The drug that he used was hydrocodone, which is the active ingredient of Vicodin. When he used water instead of Vicodin to pump into the platform, what he found is that at both the active and the inactive platforms, the fish would explore and trigger an event at the platform about 800 times on the first day. And then by the 5th day, they become less curious and less exploratory. And so they trigger activity at both platforms equally, about 300 times. But when he used Vicodin or hydrocodone instead of water, this is what he saw. By the 5th day, the fish have lost complete interest in the inactive platform, and instead they're swimming over the active platform about 2,000 times a day. I'll just show you what this looks like here in this video. So this is this arena, and you can see that on the 5th day, the fish have lost all interest in this white platform, and they spend all their time swimming over the yellow platform. In fact, each fish may wander away for a few seconds, but it comes back within a few seconds to swim over the platform and receive another dose. Now, clearly something is going on here, and if we pre-treat the animals with naloxone, then they stop this behavior. But it's possible that this is just a passive association, that the fish have started to associate this area with opioid, and so they like being in that area, and they're not actively seeking a drug. And so it was important for us to figure out whether this is passive or active administration. And one of the best ways to do that is something called a progressive ratio test. This is a protocol where you take a conditioned animal, and then although it's learned that every time it swims over the platform it gets a dose, now you change the ratio so that it has to swim over the platform multiple times to get a single dose. And if the animal is just associating that area with the drug, then their behavior won't change. But if the animal is actively seeking the drug and has made an association between that operant task of swimming over the platform and receiving the drug, then the fish will increase the number of operant actions that it performs to compensate for the increasing ratio. And so we tested these conditioned animals with requiring them to swim over 1, 5, 10, 15, or 20 times to get a single dose of drug. And this is what we found. So normal fish will swim about 2,000 times over the platform to get a single dose. But when you change the ratio to 5, 10, 15, or 20, you can see they ramp up the number of times that they swim over the platform to compensate for that changing ratio. And this provides evidence that they've really associated the act of swimming over the platform with receiving a dose of opioids. I won't go through, in the interest of time, all the details, but we found that these opioid-conditioned fish, they will engage in risky behaviors to seek a dose of opioids. When you deny them the opioids, that they go through signs of withdrawal, and that naloxone and other drugs that are known to block drug seeking in humans also prevent the zebrafish from pursuing opioids. So obviously what we're trying to do now is to screen for new drugs that might be able to prevent drug seeking in conditioned animals. And so here we've taken heavily conditioned animals and screened them against about 100 known drugs. And we found, so this is an untreated animal, has a strong preference to the active platform over the inactive platform. Compounds that have no impact look like this. They still retain that preference for the active platform. But we've been able to find a couple of new compounds that seem to block the desire to seek opioids in highly conditioned animals. Let me just end with 2 slides now that highlight what one of the main reasons why I came to the University of Utah. As you all likely know, there's a really rich resource here of genetic information that through the Utah Genome Project and other efforts is identifying dozens of new disease-causing genes in humans. We're really excited about the prospect of being able to take those new genes, validate their function, and perform functional studies in zebrafish, and then very quickly move those fish into high-throughput screening to identify therapies that can treat these diseases that are coming out of the Utah Genome Project and other fundamental studies that are happening here at the U. So the idea here is to take normal fish, use genome editing tools like CRISPR-Cas to make genetic models of these diseases, and then within a few months be able to get them into a screening platform where we can find new drugs that suppress those diseases. And that's going to be part of a big effort that we're trying to build out in the College of Pharmacy. So I hope I've mentioned some of these people along the way. I want to acknowledge Chaz Hong, who really initiated the BMP project. I mentioned most of these collaborators and support from the NIH Trend and Bridges program. The heart failure program was really led by Yan Liu with help from a cardiology fellow in the lab, Arthi Asnani, and our collaborators. And Dave Coquel, Andrew Rennekamp, and Gab Bosse were really drivers of the opioid story and some of the other behavioral screening that we're doing in our lab. So hopefully some of this has been of interest to you, and if you have any questions, I'll be delighted to try and answer them.
That was an awesome talk. 2 questions. The first, I have to ask, what's risky behavior in a zebrafish? I have to know. And then the second question with the visnagin, is that how you say it?
Yes. Yeah.
Do we understand what the mechanism is, actually? Or are we closer to knowing that?
Great. I'll try and address both of those. In terms of risky behavior, it's not using dirty needles. It's not unprotected sex. It's what fish don't like to do is be eaten. And so typically a zebrafish will not swim into really shallow water if they can avoid it. And so we've been able to then test where we elevate these platforms so that they have to swim into really shallow water. That would subject them to predation and something they normally wouldn't do. Unconditioned fish won't do it, but once you've conditioned them to seek opioids, then they'll swim up onto that platform. So in terms of visnadin, the mechanism of action is something we're working on, and we've found actually two things so far. We took that compound, we attached it to solid support, and pulled out binding proteins. And there's a metabolic protein called MDH2, which is part of the malate aspartate shuttle. And what we think is happening is that when you treat with doxorubicin, NAD levels plummet. And a lot of tissues can tolerate that, but metabolically active tissues like the heart can't. When we co-treat with visnadin, it's somehow perturbing the activity of this malate aspartate shuttle, which shuttles NAD across the mitochondrial membrane, and it restores the NAD levels. And we think that that's how it's able to protect these metabolically sensitive tissues like the heart. We're also finding, though, that these compounds are potent inhibitors of some of the metabolic enzymes that break down doxorubicin. So CYP1, for example. And we think there might be also a contribution that dox is being converted to a toxic metabolite that may also be impacting the heart, and that by blocking the metabolism of doxorubicin, you can have less of this toxic metabolite and therefore less toxicity. So we're not sure what the balance of these two mechanisms is, and we're trying to tease that out right now. Great questions, thank you.
Thank you so much. My name is Lindsay Mann, one of the heart failure cardiologists. So the doxorubicin is a very hot topic. It's near to our hearts. I just wondered if you gave the protective agents, were they given for primary prevention before the fish were exposed to doxorubicin, or did you give them after they developed heart failure and then you saw reversal of it?
Yeah, great question. If we wait a day or two after the doxorubicin exposure, there's no protection. So this has to be present at the time that the animals receive doxorubicin to have an effect, which would be consistent with both of these mechanisms that I mentioned.
Tell us about the screen. How do you select?
Yeah. So the libraries that we use depend a little bit on the screen and what our throughput is and what we're trying to accomplish. We have collections of FDA-approved drugs, and those are often a good starting point, especially when therapy is the end goal. We'd love to be able to find existing drugs that have already been tested for safety that could be repurposed for a new indication. But we often exhaust those and don't have something compelling, and so we move to then larger chemical compound libraries. These are collections that have been synthesized by armies of chemists with no real rationale, just making what they can make. We have them arrayed in 96-well format, and so when we fill a plate full of zebrafish and then we stamp in the compounds from these libraries, put them in our screening systems, and when we identify a hit, C7, you sunk my battleship kind of thing, then we can go back to the database and figure out what that compound was. Generally, nothing is known about those. They're just, novel structures, and that's when kind of the fun begins of optimizing them and figuring out what their mechanism of action is. Yeah.
Good morning. Thanks for that talk.
Morning.
I am from a more macro level, less pathophysiologic, thinking about resource utilization, and when you're going after these orphan diseases, how does that work on a national level so that there aren't a bunch of labs going after the same rare diseases, and how do you sort that out? Maybe it's a naive question, but I'm just curious about that.
Yeah, it's not a naive question, and it's an area where there's been fairly poor coordination. About 5 years ago, no one was interested in FOP. Once we developed some of these compounds, we've now licensed them to two companies, and there are another maybe 3 companies who've got their own programs going in FOP. So we now have almost as many companies developing FOP therapeutics as we have patients. So I think better coordination would be really helpful, and to my knowledge, it's not being coordinated very well.
Any last questions? No? Okay, thank you.